morning to all. Welcome to the Diamond Group of Institutions NEET and K-CET online classes. Today's topic it is anatomy of flowering plants. Study the given flow chart. So in this, they given as a flow chart. The flow chart represents as a different types of tissues. The different types of tissues are mesomatic tissues, simple permanent tissues, and complex tissues. Based on this, they made a flow chart. See now here, cells in a continuous state of division. Cells in a continuous state of division that represented as a mesomatic tissues. Which are the cells are that it shows the continuous divisions. That cells we call as a mesomatic tissues. So if these are mesomatic tissues, we can call as it is a mesomatic tissues. If we can say as it is, that is labeled with P. So here P represent as P can be root apical meristem, which is generally subterminal position. If this is S, we can consider as a P. And if those are not a mesomatic tissues, they given as a no. They given as other one made of similar type of cells. If these are the tissues are not mesomatic tissues, we call as a no. Made of similar type of the tissues, we call as a tissues only, that is permanent tissues. Permanent tissues are again classified into two types: simple permanent tissues and complex permanent tissues. So if we are called as these are permanent tissues made of similar type of cells, if we call as a no, means we call as these are the complex permanent tissues. The complex permanent tissues contains only one type of the dead cells. The complex permanent tissues are we call as a xylem or phloem, either. So xylem can contain the water, phloem can contain the water food materials. So here, if contains only one type of a cells or dead cells, that is we call as a S. They may label with Q. Q they given as Q can be phloem, which is also called as a bast. And next, here these all are the tissues these are the simple permanent tissues. We can call as simple permanent tissues. Again, S chiefly mechanical tissues. If those are called as a simple permanent tissues, chiefly mechanical tissues means they can provide the mechanical support. So if we are called as a no, they can label as a or can be parenchyma, which comprises a thin wall isodiametric cells. And if this is not a mechanical tissues, or it says, if it is a mechanical tissue, it says as possess uniform lignified walls. If that is possess the lignified walls, that is called as a dead cells. If it is S, it is called as a S. So based upon that, by the using of the flow chart, it says as the different types of the tissues, the different types of tissues are parenchyma, collenchyma, sclerenchyma as a permanent tissues, and mesomatic tissues, and complex tissues as a xylem and phloem. So you know these are the complex tissues and the mesomatic tissues and the permanent tissues. Based on that, which of the following statement is incorrect? Out of the given options, we have to identify the incorrect statements. So the incorrect statements: P can be root apical meristems, which is generally subterminal position. If you are considered as this is the P it is present, considered as a mesomatic tissue. It is present generally in subterminal position. Yes, here mesomatic tissues are that it shows a continuous division. We are called as this is a mesomatic tissue that is labeled with P. So means that P are located at the subterminal position, means they are present at the apical region. We call as that apical meristems. That apical meristems as root apical meristems and shoot apical meristems. Means the given statement A is correct only. And option B, Q can be phloem, which is also called as a bast. Here now you see this is the Q. If these are the mesomatic tissues are not considered, means made up of similar type of cells, we call as a permanent cells. Those are the permanent tissues are not. So they are given in two, means we are called as a simple permanent tissues and complex permanent tissues. So these are the simple permanent tissues. This is says as a simple permanent tissue. This says as a complex permanent tissue. It contains only one type of the dead cell. The dead cells are we are seeing into the xylem and phloem. So here, if it is S, they mention as a Q. Q can be phloem, which is called as a bast. Now we are called as it is a phloem. Phloem contained as a bast cell. So option B also correct. And option C, R can be parenchyma, which comprises of the thin wall isodiametric. See now, here these are the simple permanent tissues. Option C, they are given as a chiefly mechanical tissue. Chiefly mechanical tissue. The chiefly mechanical tissues are we are seeing as a sclerenchyma. So those are the not sclerenchyma cells. See now, this is the statement belongs into the sclerenchyma. If this is a sclerenchyma, it will be going continuously to yes. If this is not a sclerenchyma cell, means the cell will be going to living. The living cells are parenchyma and collenchyma only. So now it is belongs into the R. R says as R can be parenchyma, which comprises the thin walled isodiametric. So now this is the R parenchyma cell. So R will not made up by the giving as a mechanical tissue. So option C also correct. Now see now option D. S can be collenchyma, which is living mechanical tissue. If you are taken as a chiefly mechanical tissues, we are called as it is a chiefly mechanical tissue sclerenchyma. It is S possess uniform lignified cells. Sclerenchyma cells are cell wall made up by the lignin, so they can provide into the mechanical support. If that is called as considered as a S, they given as a S. S can be collenchyma is living mechanical tissue. Now here S it is belongs into the sclerenchyma. It is a sclerenchyma. It's a non-living tissue. But in the option D, they given as a S can be collenchyma. Here only S should be present because complex tissues are simple permanent tissues and complex permanent tissue is in the simple permanent tissues. Parenchyma, collenchyma, sclerenchyma is there. So in these are the three. These are the parenchyma does not provide as mechanical support. Collenchyma, sclerenchyma they can provide the mechanical support. Here we will be taking to the living mechanical tissues as a collenchyma, but the collenchyma cells are not made up by the lignified cells. If the cells are made up by the lignin, that is belongs into the sclerenchyma. But here see S can be collenchyma, which is living mechanical tissue. S can be collenchyma, which is living mechanical tissue. Yes, it's not a collenchyma, it is a sclerenchyma. So it's not a living mechanical tissue, it's a dead mechanical tissue only. Incorrect statement it is D, not a B, C. These are the A, B, C are correct statements. And next question. Which are the following possess nucleus? Means which are the cells are they contain as a nucleus? Options are they given as vessels, sieve cells, xylem parenchyma and tracheids. Means out of this all, which cells are they contain as a nucleus? So now vessels and the tracheids are the non-living cells. Means they won't have the nucleus. Nucleus is absent. And remaining the sieve cells and the xylem parenchyma. Sieve cells and the xylem parenchyma. So sieve cells and xylem parenchyma cells both are living, but sieve cells are living. These sieve cells will be going to maintain by the collenchyma cells. So collenchyma cells are having as a nucleus, but the sieve cells they won't have any nucleus. So remain as a xylem parenchyma. Xylem parenchyma is a living tissue. It is contained as a nucleus and is helpful for storage of the food material. So out of the given options, nucleus presence in xylem parenchyma only. Answer is C. Next question. Sequence of cellular layers from the periphery towards the cortex in an old dicot stem. We have to identify the sequence of layers present in a old dicot layer, that is from the periphery towards the cortex. Means from the outside to inside, we have to identify the sequence of the layers towards the cortex, otherwise towards the vascular bundles. So that is in a dicot stem. So you know that always outermost layer is epidermis, below the epidermis. This is the epidermis. So option A, epidermis, hypodermis, phyllogen and phylum. So see now, old dicot stem. In a dicot stem, hypodermis is absent. So option 1 means option A is incorrect. And coming to the option B, epidermis, phyllogen, phylum and phylloderm. So below the epidermis, they given as a epidermis, phyllogen. Phyllogen means cork-canium. Actually, below the epidermis, here they presents as a phylum, later phyllogen, and later this is phloem, and later this is endodermis, and later here phloem, later cambium, later here xylem, and here it will be going to present. This is the arrangement of the layers. See now option B, epidermis, phloem they given. Here phloem, it's not a correct, and it is also called as a pod cambium. Before the phloem, it will be going to present as a pod. So option B also incorrect statement. And option C, epidermis, hypodermis. Here again they mention as a hypodermis. Hypodermis is a character of monopod. So option C also incorrect. Now you see epidermis, outermost layer as epidermis. Next phloem, below the epidermis, phloem cells we are also called as a pod cells. And below the pod cells, phloem we are also called as a pod cambium. And later phloem we are also called as a secondary pod. So this is the sequence of layers arranged from the outside to inside in a dicot stems. So answer is D. And next question, which one is not the characteristic of meristematic cells? So you know that meristematic cells are the cells, they are present in all living plants. They are specialized tissues, they can show continuous divisions. Meristematic tissues means meristematic cells are that shows a continuous division. So based on the meristematic tissues, option A, compactly arranged cells without intercellular spaces. Yes, meristematic tissues cells are there arranged compactly. They are arrangement take place closely without having any intercellular spaces. Yes, here the intercellular spaces are absent. And next option B, walls thin, elastic, and made up of by cellulose. Yes, these are thin wall cells. Thin wall cells. Cell wall it is made up of by cellulose and elastic nature. Elastic means expandable, and large amount of ergastic substances are present. So now you see large amount of the ergastic substances are starch. So these are the large amount of ergastic substances they can stored in parenchyma cells, or they are also called as a permanent cells. So storing of the food materials is a character of permanent cells, or that too in a permanent cells as a parenchyma. So, and option D, these are immature and undifferentiated cells. Yes, these are the immature cells only. When they are called as a mature cells, that is called as a permanent tissues. Those are immature cells and undifferentiated cells only. So out of the given options, incorrect statement is C, large amount of ergatic substances. They can store the large amount of ergatic substances in that statement. And next question, read the following statements round 1. We have to identify the incorrect statement of the given below. Option A, gymnosperms have a albuminous and sieve cells in their phloem. Yes, gymnosperms are the after the completion of the pteridophytes, gymnosperms are then shows the vascular tissues. In that vascular tissues, albuminous cells means instead of the companion cells, albuminous cells are will be going to present. Instead of the sieve tube cells, sieve cells are there present in a phloem. Yes, and option B, the cell wall of parenchyma made up of cellulose, hemicellulose, and pectin. Parenchyma is a living mechanical tissue. It can made up of by the cellulose, hemicellulose, and pectin. So option B also correct one. And option C, the first formed primary xylem elements are called protoxylem. So first formed xylem cells are we call as a protoxylem. Those are having a small vessel, and after formed xylem cells are we call as a metaxylem. They given as a first formed. First means proto. It's called as a protoxylem. Next. Option D, the cell wall of parenchyma is made up of by lignin. So they are asking the round one. So here the cell wall of the parenchyma is made up of by cellulose, but they given as a lignin. So incorrect statement is D only. And next one, vessels in xylem are found in. Vessels, presence of the vessels is the character of angiosperm plants. So those are the vessels we are seeing in option A, all angiosperms and gymnosperms. All angiosperms and gymnosperms. If you are consider as all angiosperms, some of the angiosperms are vessel-less angiosperms. Vessel-less angiosperms. Those are Rhododendron and Dryness. So these are the two plants are Rhododendron and Dryness, vessel-less angiosperms. Means in the angiosperms of these two vessels are absent. So if you are taken as all angiosperms they are having vessels, that option it is a incorrect. So generally vessels are the specialized tissues. They are involved into the transporting of water and minerals. So the presence of the vessels is a character of angiosperms, but all angiosperms does not contain the vessels. Then exemptions are these two. And gymnosperms. Gymnosperms are here. Gymnosperms are they won't have the vessels, but some of the gymnosperms, some of the pterophytes vessels are their presence. Those are the gymnosperms are Selaginella. Pterophytes are Selaginella and Ephistum, and gymnosperms are Neetum. So this is the Neetum gymnosperm. It's contained as a vessels. Selaginella and Ephistum they belongs into the pterophytes. So all angiosperms and gymnosperms they won't have the vessels, and most of the angiosperms and few gymnosperms. So most of the angiosperms, presence of the vessels is a character of angiosperms. These two only exemption. Except these two remain, all of the angiosperms are they have the vessels. So most are angiosperms and few gymnosperms. That few gymnosperms are neat. And option C, few angiosperms and all gymnosperms. Here most of the angiosperms and some gymnosperms. And option D given as all pterophytes. That correct wrong statement is we have to identify the presence of vessels in most of the angiosperms and few of the gymnosperms. Answer is D. Next question, 7th one. Leaf of dicot differentiate from monocots by having. So here we have to differentiate the dicot leaf with monocot leaf. The dicot leaf having. We have giving a comparisons of dicot leaf with monocot leaves. By that comparisons also the dicot leaves are having. Option A, parallel venation. See, this is the difference between the dicot leaf and monocot leaf. But parallel venation is a character of monocot. And stomata only on upper epidermis. So this is also the difference between dicot leaf and the monocot leaf only. But stomata only on upper epidermis. Stomata only on upper epidermis. It is a character of monocot. And differentiation of mesophyll cells into palisade and spongy parenchyma. Now you see. So here when you cut the transverse section of the leaf of dicot, this is the vascular bundle. This is called as a mesophyll cells. In the dicot leaves, the mesophyll cells are classified into two types as a palisade parenchyma and spongy parenchyma. So differentiation of mesophyll cells into palisade and spongy parenchyma. Here in the dicot leaf only such type of differentiation we can see. When you cut the transverse section of monocot leaf, the monocot leaf entirely filled by the spongy parenchymatous tissue only. Palisade parenchyma is absent. So the differentiation of mesophyll cells into two types of the cells is the character of dicot leaf. The two types of the cells are palisade parenchyma and spongy parenchyma. And option D, sclerenchymatous bundle sheath extensions. That sclerenchymatous bundle sheath extensions on the surface of the vascular bundles of monocot leaf, a sclerenchymatous bundle sheath extensions are we are seeing. Dicot leaves they won't have any sclerenchymatous bundle sheath extensions. So monocot and dicot leaf differences. In the differences also, which is having dicot leaf, the differentiate into the monocot leaf. See now, parallel venation is a character of monocot. Stomata only on upper epidermis is a monocot. Differentiation of mesophyll cells into a palisade and spongy parenchyma. So now these are the palisade parenchyma and spongy parenchyma. The polycell parenchyma and spongy parenchyma, we are seeing a didot leaf only. It is a difference of monodot leaf also. So answer it is C. And next question. Anatomically, dicotyledonous root is distinguished from the dicot stem. So the major differences between the dicot root and the dicot stem, you can see as this is the root and this is the stem. So in the dicot root, it's a tetrarch. Vascular bundles are there present in the form of tetrarch. In a dicot stem. So differences between the dicot root and the dicot stems. Options are absence of secondary phloem. Absence of secondary phloem. Here both are the dicot root and dicot stem that can perform the secondary growth. So both are that can show the secondary phloem. And here option B, absence of vascular cambium. Absence of vascular cambium. See now, absence of the vascular cambium also. Here in the dicot stem and the dicot root, vascular cambium it is present because dicot root and dicot stem both are they can perform the secondary growth because of the presence of vascular cambium only they can perform the secondary. And position of protoxylem. Now you can see position of protoxylem and roots and the stems. Arrangement of the xylem cells. Arrangement of the xylem cells. So xylem cells are first formed xylem cells we call as a protoxylem. Later formed xylem cells are called as a metaxylem. So this is the protoxylem and this is the metaxylem. That is in roots. But when you see in the stems or shoots. So this is the vascular bundle, this is the vascular cambium, and here now you see large size xylem cells are we call as a metaxylems and small size xylem cells are we call as a protoxylem. So this is the protoxylem, this is the metaxylem. Position of protoxylems and metaxylem in a roots and stems are they will be going to difference. So in a roots this is called as a exact vascular bundle. In a stems, this type of the arrangement we call as a endark vascular bundle. So now, anatomically dicotyledonous roots and stems are differentiated as absence of secondary phloem, it is not correct. Absence of vascular cambium, this is also not correct. And position of protoxylem. In that roots, protoxylem it was present at periphery, metaxylem at center. That is called as a exact. In a stem, protoxylem it was present at the center, metaxylem it was present at the periphery. Based upon the position of the protoxylem, here exact means outside, here the endark means that protoxylem it was present at the center. So the major difference between the root and stem it is the position of xylem cells we can identify. So answer is C. And option D they given as absence of secondary xylem. So secondary xylem in a dicot stems and dicot roots both are they can show because they can perform the secondary growth. And next question, axillary bud and the terminal bud are derived from the activity of. Axillary bud and terminal buds are derived from the activity of. The question it is axillary bud and terminal bud. Axillary bud and terminal bud. So here the bud will be going to develop. This is called as a terminal bud. Here the bud will be going to develop. This is called as a axillary bud. See now, options are axillary bud and terminal bud are derived from option A, lateral meristems, intercalary meristems, apical meristems, and parenchymatous tissue. See now, we based upon the position of the mesomatic tissues. Mesomatic tissues are classified into three types. Those are apical, intercalary, and the lateral. See here the lateral meristems means mesomatic tissues are they present at the lateral sides of the plant body. So these are we call as a lateral meristems. And intercalary meristems, mesomatic tissues are present at the nodal region. Those are we call as a intercalary meristems. Apical meristems, Meristematic tissues are there present at the tip of the plant body, we call as a apical meristems. And parenchyma tissue. See now here, they are asking terminal bud. Terminal means apex region, and the axillary bud. So the axillary bud will get develop into the intercalary meristem. So now here, both are the terminal bud and the axillary bud. They are developed from the apical meristems and the intercalary meristems. See now here, apical meristems and intercalary meristems are given. Here the intercalary meristems are also getting developed here. Axillary bud will be developed in between the leaf and the stem. So now these are the axillary bud will be development take place from the apical meristems only. Here apex region, here meristematic tissues are present, and here also the axillary bud will development take place from the apical region only, because of these first meristematic tissues are there present. They do the formation of bud, they are called as apical meristems only. Lateral meristems are used in a secondary growth. Intercalary meristems are there present in nodal region. Parenchymatous tissue will not show. So remaining answer is C only. And next question 10. Read the following statements with few blanks and select the correctly. Fill the blanks in two of them. So they given as some options with the blanks. Out of the two blanks, we have to identify any two. Option 1. The center of the monocot root occupied by. The center of the monocot root here, when we draw the diagram for this, center of the monocot root here, this is. The center of the monocot root is occupied by the pith. Occupied by, they are given as a dash. They are going to represent as the option 1 means pith. And option 2, in dicot roots, secondary phloem is made of seed tube cells. Here, in a dicot roots, secondary phloem is made of seed tube cells, and they are given as a seed tube cells. And here in the second option, the branch will be given as a companion cells. Seed tube cells, companion cells, and phloem parenchyma. And option 3 they given as the medullary rays are helps in the conduction of. The medullary rays are helps in the conduction of the food and water. Yes, the medullary rays are helps in the conduction of the, that is they are helps into the conduction. The conduction is usually going to present as a radial conduction. And option 4, the epidermis. The epidermis of sunflower stem bears 2. The epidermis of sunflower bears 2. They are the epidermis of sunflower that will bears into the hair-like structure. That hair-like structures are called as a rhizomes. So now you see, out of the given 4 options, again in the given as options. Option A, cortex. If you are considering as a option 1, it's correct. Option A 1. The center of the monocot is occupied by cortex. It's not a cortex, it's a pith. And option B, 3 is a radial. Yes, 3 is a radial. So in dicot roots, secondary phloem is made up of by. The medullary rays helps in the radial conduction. Yes, 3 is a radial conduction, and the 4 they given as motor cells. Means 4 what they given, the epidermis of sunflower bears into the motor cells. Here, motor cells are the bulliform cells are we are seen in monocots of upper epidermis. So here B also incorrect statement. And option C, 1. The center of the monocot root is occupied by the pith. And option 4, the epidermis of sunflower bears the rhizome. So option C 1 is a pith, and option C 4 is a rhizome. So 1 pith and 4 rhizome. Answer C is a right. And C again option D, sclerenchyma and axial. So option 1 they given as a sclerenchyma. It's not a sclerenchymatous tissue. It is occupied with the pith, and it's not an axial. It's a radial conduction of water. So answer it is C.